Today's presentation is how to define and measure clinical endpoints to optimize your oncology drug dosing. Our speakers are Dr. Julie Bullock, Dr. Vincent Duval, Dr. Italo Poghesi, and Dr. Adekemi Taylor. Julie is currently a Senior Vice President at Certara, where she heads up the Global Clinical Pharmacology and Translational Medicine Group. Vincent is currently a Senior Director at Certara in the Quantitative Science Services Group. He's joined by Italo, who's also a Senior Director at Certara in the Quantitative Science Services Group. And finally, we've got Kemi, who's a Vice President of Pharmacometrics in the Eastern region of the United States in the Quantitative Science Services Group. Welcome to today's webinar. I'll now turn it over to Julie to begin the presentation.
Great. Thank you, Suzanne, for that introduction. In my talk today, we will walk through the available endpoints for oncology, and I will discuss the difference between informative endpoints and surrogate endpoints, as well as the differences between non-validated and validated surrogates. I'll then discuss how to use various endpoints through the dose decision-making journey and how they can be leveraged. Lastly, I'll go through some quick examples on how to integrate these endpoints for dose selection. Next slide. When putting this talk together, I find it easiest to bucket our endpoints, and I came up with 3 buckets: clinical benefit endpoints, surrogate endpoints, and what I'll call informative endpoints. Clinically meaningful endpoints relate to the outcomes which capture how a patient feels, functions, or survives, and these endpoints may be measured objectively or subjectively. The gold standard clinical benefit endpoint for oncology would be overall survival, but since clinically meaningful effect for the purposes of the Food and Drug Administration is defined as how the patient feels, functions, or survives, quality of life is another endpoint that could be used, although is rarely leveraged for clinical benefit purposes alone for oncology. The next bucket is surrogate endpoints. These are endpoints such as progression-free survival, overall response rate, durable CRs, and some of these endpoints could also fall into the bucket of clinical benefit endpoints, depending upon if the surrogate has been validated. Next slide. For surrogate endpoints, there are non-validated or intermediate surrogate and validated surrogate endpoints. An intermediate surrogate is an endpoint that is reasonably likely to predict clinical benefit, and these endpoints are often used for accelerated approval as they are endpoints that can be collected earlier than long-term endpoints like overall survival or PFS. For non-validated or intermediate surrogate endpoints, additional follow-up is needed to confirm benefit. And some examples of intermediate surrogate endpoints would be durable overall response rate for many solid tumor indications or pathological complete response for breast cancer. In addition, there are also validated surrogate endpoints, and these endpoints are known to predict clinical benefit and can be used for traditional or regular approvals. Not all surrogate endpoints have been validated to replace the need for overall survival or PFS. An example of a validated surrogate endpoint is durable major molecular response for CML. Next slide. The last bucket I'll talk about is informative endpoints, and this includes biomarkers, which do not relate directly to how a patient feels, functions, or survives, but are instead objectively measured indicators of a biological or pharmacological response to a treatment intervention. Biomarkers may include pharmacokinetics, circulating tumor DNA, blood tests or tissue samples, as well as imaging results. These informative endpoints can assist in understanding the characteristics of an asset, but alone are not sufficient to support clinical decision-making for approval purposes. Their utility for dose optimization is important, and we will come back to that in a moment. Next slide, please. Before I move on, I wanted to touch a bit on the goal for Project Optimus. In short, the goal is to enhance the dose finding and dose optimization approach across oncology to evaluate a selection of doses that maximizes efficacy in addition to safety and tolerability. So the FDA wants us to change the paradigm, but what is the goal of a dose finding study? The goal of dose finding from the Friends of Cancer Research November 2021 white paper was to adequately characterize the exposure safety and exposure activity relationship. While I agree these goals are ambitious, I think the advice here is rather pragmatic and flexible. Next slide, please. So when would we collect this data? I presented this finding of a conceptual framework in May at the FDA ASCO Project Optimus workshop. And after months of working with clients in the field, I still stand by the premise that dose optimization occurs in two stages that can often blur. As I walk through this slide, it's important to know that stage 1A to 2 can easily be accomplished in one study, or it can be split into two. And this would be a development decision and would depend on the sponsor and the asset. Even the efficacy component can be wrapped into a single study with the earlier stages, but for purposes of illustration, I've delineated it as separate. A key takeaway from this framework is that it's very rare for our standard first-in-human dose escalation studies conducted in oncology to provide enough activity and information to guide what doses should be taken forward for further testing. Therefore, additional patients will need to be studied, and this is depicted by stage 1B, which could be done via backfill and escalation cohorts or through expansion cohorts, looking at multiple dose levels. This early data will then help, along with non-clinical pharmacology data, to determine an active dose range for further efficacy testing. Hard and fast rules on how we progress through stage 1 and 2 will be difficult to outline, but for sponsors, the ultimate goal we are trying to achieve is to capture broader data across the dose range before determining a dose for registrational efficacy studies. Next slide, please. The endpoints for dose selection will also change as we go through the data gathering journey. In early drug development, we will be relying on the informative endpoints to narrow the range of doses we want to gather additional efficacy data on. These informative endpoints are not going to be robust enough to definitively determine the optimal dose for registration, but they can help in assisting in narrowing the field of viable dose levels. Next slide. Once we have gathered enough data, our decision for determining an optimal dose should be based on endpoints that are more surrogate in nature, endpoints that are reasonably likely to predict clinical benefit or are validated to predict clinical benefit. But efficacy alone is not the only endpoint to making a dosing decision. We must also look and consider at other informative endpoints such as pharmacokinetic, safety, pharmacodynamics, in addition to efficacy. Next slide. Now I'd like to walk through a few case studies. Torcel is one of the drugs I worked on while I was at the FDA, which had a legitimate dose-finding data which contributed to its approval. In addition to PFS being established across the dose range, early readouts on overall response showed in the bottom two graphs, showed a lack of a dose response, which supported 25 milligrams weekly dose, even though the overall safety profile across this 25 to 250 milligram dose range was rather innocuous. Additional data that often isn't brought up in the Torcel discussion, though, is the early biomarker data that was gathered in healthy males that showed that a dose response for target coverage across the dosing interval that was saturated at 25 milligrams. Note, this biomarker data was not used to as a definitive endpoint for dose selection. It was used as an informative endpoint to bracket the lower efficacy dose the sponsor took into dose ranging efficacy trials. Next slide, please. Pembrolizumab is another case example that we will dive into a little bit deeper later in this webinar, but it's a great example of how informative endpoints were used early to narrow the range of clinical efficacious doses. In this example, the ex vivo IL-2 assay, along with modeling and simulation, provided that the lower threshold for efficacy for Pembro is likely achieved at a dose of 2 mg per kg Q2 weeks. And while additional higher doses were evaluated clinically, the exposure response of efficacy and safety endpoints showed no difference between either response or safety at doses ranging from 2 to 10 mg per kg, providing further support of the 2 mg per kg dose. Notice in this example that Pembro did not use the informative endpoint, ex vivo IL-2 assay, to definitively define the dose for Pembro. They used it to narrow the range of doses and provided clinical efficacy data to justify the optimal dose. Next slide. As I wrap things up, in the Friends of Cancer Research White Paper, an outline of the 4 key areas and associated critical questions for supportive evidence of the proposed dose was outlined. I provide a brief summary of the table here and have highlighted that for all of the key areas for translational, PK, efficacy, and safety, that dose or exposure response relationships are being scrutinized. Next slide. In conclusion, for dose optimization, there is no singular endpoint that will be used for decision-making. Not only will the decision-making endpoints change through development, but there are multiple factors that need to be considered for determination of an optimal dose. The first goal we must all commit to is to first collect the data. Collect a variety of data and endpoints across an active dose range in a relevant population, and study these data and endpoints in a meaningful number of subjects. Secondly, interrogate this data. Does the pharmacokinetics increase? Do I see a plateau in the pharmacodynamic response? Is there a difference between time to response among doses, or is the duration of response different? Is the time and intensity of adverse events, is the dose intensity different across the dose range, etc.? And lastly, integrate this data for storytelling. Leverage model-informed analyses to inform decisions and simulate alternative scenarios. It is important to note as well that these data analyses activities need to be planned for in the drug development timeline. These analyses take resources and time and should not be overlooked when planning for health authority meetings and planning decision-making timelines. Slide 15. Or next slide, sorry. So thank you for your attention. I'll hand it over to Vincent, who will talk a little more about model-informed development and simulation.
Okay, thank you so much, Julie, for this nice presentations on the biomarkers and how they can be used for those selections in oncology during the drug development process. Together with Italo and Kemi, we will drive you now through a few examples to show you how modeling and simulation can eventually help and through different programs. Why do we want to do this? Is maybe because you all realize that recently FDA raised the bar in terms of those selections in the context of oncology, and that currently MTD approach is maybe not appropriate any longer. So we believe that modeling and simulation can really be a proper tool to use appropriately the endpoints, the early endpoint available in early phase of development. So what we recommend is really using modeling on an ongoing basis as early as possible during drug development, knowing the challenges that we have during development. This means heterogeneity of the population in the early phase, limited number of subjects during the dose escalation phase, and collection of relevant information that can be difficult. So, I mean, modeling and simulation really can help leveraging this limited information in terms of biomarkers, efficacy, and safety. Just maybe to set the scene and show you one example of feedback that we recently received from the FDA and emphasizing the importance of modeling and simulation as well as drug exposure response. This is a situation of a client who wanted to start an early phase 3 studies and consulting FDA. Then, based on the briefing book they submitted, they received the following answer. Please provide integrated dose exposure response analysis using preliminary and emerging efficacy and safety data. FDA also wanted to see tabulated summaries for PK, for PD data, safety data, preliminary efficacy data. And they also wanted to see population PK and exposure response analysis for the study protocol. So this means that if you're not prepared to answer such questions without the data, with collecting the data, then I mean you can end up being in difficult situations. So to move on with this presentation, we divided this presentation in 3 stages where we think there is important milestone that can help, where modeling can help. The first milestone that will be presented by Italo is the transition between preclinical stage and early stage, where the main question is definitely defining the safe starting dose and establishing a proper protocol design that can help answering the questions during the phase 1 and phase 2 study. What is a proper protocol design? We're talking about PK samples, we are talking about proper biomarkers to collect, and a plan to analyze the exposure response. To address this type of questions, we know that we can use translational PK/PD modeling to make the protocol design more appropriate and being more efficient in using our data from phase 1 and phase 2. Once we establish the phase 1 protocol, then we start collecting the data. Which type of data are we collecting? We will collect early efficacy readouts. This will be informative biomarkers, as Julie mentioned earlier. It will be PFS, it will be safety signal. So this endpoint will be used for confirmation of the dose, confirmation of the indication, confirmation of the pharmacological activity, and potentially combination. How do we want to use these early endpoints? This can be as well, that's where as well modeling can be used to define and develop early PK/PD models using biomarkers, using tumor size modeling, and potentially PFS, which is an early readout. Then the last challenge is how do we relate these early endpoints with the clinical endpoints that is of interest for us, which is often quality of life or overall survival. And this is where we can as well use modeling, using models relating tumor size together with overall survival. So to guide you through these different examples, I will first leave the floor to Italo, which will show us one example on preclinical stage, and then we will move to early and pivotal study. Italo, if you want to.
Thank you, Vincent. Can you go to the next slide, please? Yeah, we will give a look to a case example, the case example of erdafitinib in terms of continuous and timely application of modeling approach. First of all, I would like to acknowledge the former colleagues of Janssen and elsewhere who supported this program, in particular Angel Dozen and Kim Steikens, who really were instrumental to this exercise. So let's start with some background information. Erdafitinib is a small molecule, is a pan-FGFR kinase inhibitor. It was approved in April 2019 for the treatment of urothelial carcinomas characterized by susceptible FGFR genetic alteration. Erdafitinib was approved with breakthrough designation and accelerated approval based on tumor response data collected in a phase 2 study in 87 subjects with this indication. The modeling was applied throughout the whole program. It was really an accelerated program. Overall, the support from the modeling derived from 3 different ways. Essentially, the first one is anticipating the range of clinically active dose based on the non-clinical data in xenografted mice and rats. Secondly, using the data coming from the phase 1 and early phase 2 trial, providing a PK/PD model for phosphate elevation, which is an informative endpoint following the earlier definition from Julie's slides. And this was used to support both the dose regimen selection and the individualization, and eventually providing exposure response analysis of the endpoint of clinical efficacy and safety that further supported the choice of erdafitinib dosing regimen. If you go to the next slide, we'll give a look to the translational approach that was able to anticipate the potentially beneficial dose. Essentially, experiments were available in xenografted mice and rats at doses from, can you go back one slide, please? At doses from 5 to 50 milligram kilogram once or twice daily. And the PK in mice and rats was modeled simultaneously in both species using essentially an allometric correction. Tumor growth and tumor growth inhibition were modeled using a popular model, the Simeoni model, that is a standard model used for this kind of purposes. On the upper left side, there is the modeling of the PK. On the bottom left side, there is, you can see the modeling of the tumor growth curves in the different experiment. Essentially, based on this modeling, it was possible to identify a range of concentration expressed as unbound erdafitinib, above which tumor shrinkage was expected in preclinical experiment. And this range was considered the target for the dosing regimen in human subjects. In the plot on the bottom right, you can see the range of these concentrations overlaid with the PK profile simulated for different dosing regimens in human subjects. Essentially, based on the first clinical PK data, it was possible to extrapolate that doses above 4 milligrams daily would have been characterized by clinical benefit. And indeed, the first recommended phase 2 dose proposed was the 6 milligrams once daily, which provided concentration within the anticipated range of active concentration. An additional intermittent dosing regimen of 10 milligrams daily, given 7 days on and 7 days off, was also proposed from the clinician. It is interesting to notice that all the modelers supported the continuous dosing over the intermittent one. And a posteriori, it was indeed possible to justify this position using a Beck translational approach. So what we did was to essentially highlight 2 dosing regimen, scale down from human to mice, and this was used to predict the tumor growth curves in these instances. The modeling showed that continuous dosing was able to lead to tumor shrinkage in mice, whilst intermittent dosing was expected only to stabilize the tumor weight. And of note, the intermittent dosing was later abandoned from the clinical development, as it was found less active than the continuous counterpart. If you go to the next slide, we can give a look to what was the support that modeling provided to the refinement of the choice of the dose. Essentially, we had exploration of a wide range of doses from 0.5 to 12 milligrams once daily, and intermittent dosing from 10 to 12. Also, it was known based on the mechanism of action that erdafitinib was characterized by a dysregulation of the mineral metabolism, which may result in phosphate elevation, so that a PK/PD model was started immediately after the start of the clinical trial in order to have a better control, a quantitative tool to control the outcome of this informative endpoint. In the next slide, you can see essentially the PK/PD model that was used for, on the left-hand side, the PK/PD model that was used for describing the serum phosphate concentration. It is a three-compartment PK model with an effect compartment because the phosphate elevation was delayed compared to the PK. The phosphate elevation was essentially directly proportional to the unbound concentration. Other functional models have been used, but this was the best one to describe the available data. And the model also incorporated the nonlinear relationship between free fraction and individual serum alpha glycoprotein concentration. And this is described by the plot in the middle of the slides. Essentially, model parameters were refined based on the emerging observation from a phase 1 study. And the model was eventually used for a wide simulation campaign that was aiming to compare potential and different dosing regimen. If you go to the next slide, we will see some of the results of the simulation. In particular, the simulation results that we're showing here refers to the dosing regimen that was eventually proposed for the market. So it is a starting dose at 8 milligrams daily, assessment of the serum phosphate levels 2 to 3 weeks after the start of the dosing, with the potential for a dose increase up to 9 milligrams daily if serum phosphate level was not touching a threshold of 5.5 milligrams per deciliter. Naturally, those levels of erdafitinib could be further decreased based on emerging toxicity. On the left-hand side plot, the time course of serum phosphate concentration are reported for these dosing regimen. It should be noted that the simulation incorporated the effect of dose interruption and dose decreases as conditional events based on the phosphate elevation. So based on this simulation, this clinical regimen was expected to lead to elevations of phosphate minimally stretching outside the upper limit of the norm. On the right-hand side plot, instead, you can see the incidence of the different dose levels for the different time period after the start of the dose, the incidence of dose interruption, the incidence of dropouts. Based on the comparison of this simulation for all the different analyzed dosing regimen, it was concluded that dosing regimen presented here was able to minimize the incidence of dose decreases and interruptions, leading essentially to the therapeutic option which maximized the exposure to erdafitinib. And so it was used for as the proposed clinical dose. In the next slide, there is the third, let's say, portion of the modeling that was incorporated in the development of erdafitinib. Essentially, the endpoints of clinical efficacy and clinical safety were also model based on the data coming from the phase 2 trial. And again, phosphate levels was used as a metric of systemic exposure to erdafitinib rather than AUC or other plasma concentration related metrics. The exposure response analysis indicated an association between increased exposure and increase in progression-free survival that can be seen in the left-hand side plot, and also indicated the significant association between phosphate concentration and some of the toxicity endpoints, such as nail toxicity, eye disorder, central serous retinopathy, and Hansen-Foot syndrome. This analysis further highlighted the sweet spot for the to be sustained in terms of phosphate concentration to optimize the therapeutic benefit of erdafitinib while minimizing adverse event, essentially a region between 5 and 7 milligram per deciliter. It also supported the fact that for erdafitinib, phosphate level were better proxy of the clinical efficacy than standard metrics of systemic exposure. So we can go to the next slide, which is a report in the conclusion for this case study. So based on these erdafitinib case, it can be appreciated how modeling can be instrumental to set hypotheses and test them early and continuously in drug development with the aim of the definition of the optimal dose. With the translational work, the attention could be given more promptly on the range of active doses instead of losing time on ineffective doses. Quantitative models developed early can be used for model-based simulation with the aim of comparing regimens that were only explored or even not even tested at all in the clinical trials, allowing a more meaningful design of the dose regimen to be adopted in phase 2 programs. And finally, formal exposure response analysis are invaluable tools to further support the identified sweet spot for the dosing regimen. Now I'll give the floor to Kemi, who will provide additional case studies. Kemi.
Thanks, Italo. I will now present a case study on how early biomarker data were used to inform dose selection during development. We're going to consider the case of pembrolizumab, a programmed cell death receptor or PD-1 blocking antibody that was developed by Merck and company for the treatment of a variety of cancers. The mechanism of action of pembrolizumab is that it blocks the interaction between the PD-1 receptor and the PD-L1 and PD-L2 ligands. The clinical development and initial approval of pembrolizumab were largely based on a single study, KEYNOTE-001, which was a phase 1b first-in-human study of the safety and efficacy of pembrolizumab, first in patients with solid tumors and later in patients with advanced melanoma. Modeling and simulation were used extensively throughout development to support dose selection and characterize the clinical pharmacology of pembrolizumab. This included using biomarker data early on to inform the dose range to be investigated in the pivotal cohorts of the study. Inhibition of the stimulation of interleukin-2 or IL-2 in peripheral blood mononuclear cells was used as a marker of target engagement of PD-1 by pembrolizumab. So this is the biomarker data that we're going to examine in the case study. If you go to the next slide, please. The Keynote-001 study consisted of multiple parts. Initial modeling was based on data collected in Part A, which was a dose escalation part with a traditional 3+3 design, and Part A1, which was the dose expansion part. And it tested doses of 1, 3, and 10 milligrams per kilogram administered on day 1, day 28, and then every 2 weeks thereafter. And this was done, of course, in a small number of subjects. It's not shown here, but the PK of pembrolizumab appeared linear across this dose range, although there were some hints of non-linearity. A population PK model was developed for, you can still see on the same slide, sorry. Thank you. A population PK model was developed for pembrolizumab using the data collected from Part A and A1. Now the plot on the left shows the observed ex vivo IL-2 stimulation ratio, which is our marker of target engagement, and it's plotted against plasma concentration, using the circles to represent the observed data points. As you can see, the IL-2 stimulation ratio decreased at higher pembrolizumab concentrations, which represents pembrolizumab's inhibition of IL-2 stimulation. An inhibitory Emax model, which is basically a saturable model of the inhibition of simulation, was applied to these data, as shown by the blue curve. Both the data and the model indicated that saturation or near saturation of the drug effect at all 3 dose levels was evident, as we can see the IL-2 stimulation approaching 1. However, there was a large gap in the PK/PD data between the lowest concentrations and the higher concentrations. So as a result, the estimate of the IC50, which is the concentration that produces the half maximal inhibitory drug effect, was not considered reliable, and so the PK/PD relationship was not well characterized. So the data collected in parts A and A1 were therefore not considered sufficient to select the lowest dose for use in the larger patient populations in the study, and therefore more data were needed. So modeling and simulation using the data from parts A and A1 were used to guide the design of an additional cohort of the study, which would be called part A2. You can go to the next slide, please. The purpose of part A2 was to better characterize the PK and PD of pembrolizumab. Modeling and simulation using the existing PK and PK/PD models that were developed from parts A and A1 were used to assess multiple design options for part A2, and I'm showing the baseline designs here. The candidate designs included intrapatient dose escalation, with starting doses as low as 0.005 milligrams per kilogram. Because the PK/PD models had a large degree of uncertainty, the simulation scenarios accounted for possible alternative PK/PD properties to those estimated in the models, including unlikely cases, just to cover all bases. All told, there were 20 different scenarios of PK/PD properties applied to these 3 candidate study designs. Based on the simulation results, design C was selected and used as the design for part A2. If you can go to the next slide, please. The design selected for part A2 was successful in that it filled in the gaps previously present in the data, as we can see on the right. This is not shown, but the newly collected concentration time data also showed concentration-dependent clearance at the lower doses, which is typical of monoclonal antibodies. Also, we see that the part A2, like I said, was able to fill in the gaps in the PK/PD data, and so therefore these data that were collected resulted in a more robust PK and PK/PD model that could now be applied for further simulations. We go to the next slide. The PK/PD models were then updated to include all the data collected so far from parts A, A1, and A2, and the updated models were used to simulate target engagement across a range of doses of pembrolizumab. The graph on the right shows the probability of achieving 95% target engagement. So 95% target engagement represents basically 95% of maximal IL-2 stimulation inhibition at trough. And the probability of achieving it is basically just the simulated probability of subjects that are predicted to reach this target engagement level. And we see that the simulations were done across a dose range of 0.1 to 20 milligrams per kilogram every 3 weeks, shown here. The simulations predicted a high probability of saturating target engagement every 3 weeks, with every 3 weeks dosing of 2 milligrams per kilogram and higher. Therefore, 2 milligrams per kilogram every 3 weeks was tested in the pivotal cohort of KEYNOTE-001 in addition to the originally planned 10 milligrams per kilogram. And this dose has since been tested in larger randomized trials in advanced melanoma and non-small cell lung cancer. Next slide, please. The selection of the 2 milligram per kilogram dose was consistent with predictions based on other modeling efforts. The graph on the left shows simulated responses based on models that were developed from mouse data. And the graph on the right shows simulated responses based on a clinical tumor growth model that was later developed based on data from the three studies, KEYNOTE-001, 002, and 006. Vincent will tell us more about the tumor size models later. Both sets of simulations predicted near maximal efficacy at 2 milligrams per kilogram. All right, next slide, please. So now, biomarker data, while useful, are not a magic bullet, and their limitations should be understood. Firstly, peripheral biomarkers, such as those measured in blood, may not reflect the time course or magnitude of target engagement at the target site. For example, if treating solid tumors. This could be due to different concentrations and different receptor occupancy at the different sites. Furthermore, target engagement, even at the target site, does not necessarily translate to efficacy. And to reiterate what Julie said, biomarkers cannot be used for definitive dose selection, as they are only informative endpoints. Nevertheless, as demonstrated in this case study, a carefully chosen biomarker can help inform dose selection early in development. I'll now hand over to Vincent. Thank you.
Okay. Thank you so much, Kemi. And so to conclude with this seminar, I would like to drive you through the tumor size modeling and relationship to the clinical endpoint. We've seen how we can use early endpoints, and we will now see how we can use and relate tumor size modeling and with clinical endpoints such as overall survival. Maybe to start with, I will give you a short overview of what tumor size modeling is about. Tumor size modeling that relates the sum of the longest diameters, the tumor size, and that describe mathematically the dynamic of the tumor size over time. This is described with two rate constants. This is a rate constant of tumor growth that we will call Kg, and a rate of tumor shrinkage that we will call Ks, and which is dependent of the treatment effect. The treatment effect, we can relate it with a Kg or Ks depending on the concentrations or depending on the derived PK parameters such as AUC, as we will see later. To continue with the example of pembrolizumab that Kemi described earlier, the team of pembrolizumab, the development team of pembrolizumab related the tumor size modeling together with PK metrics that was AUC steady state at 6 weeks. When they were looking at the tumor size, they looked at 4 different categories. There was 4 categories of patients: non-responders, fast responders, slow responders, and biphasic responders, which were fast responders with a plateau at the end. All these 3 categories being defined based on the dynamic of the tumors for each of the individual. As we mentioned earlier, this dynamic of the tumor could be described with 2 rate constants, the tumor growth rate and the drug constant cell kill rate, tumor shrinkage rate. And the interest of such model is to see how the drug can impact both of this constant. And what the authors made is that basically they looked at the impact of AUC steady state at 6 weeks on the different metrics. And they were looking as well at the probabilities to belong at one of the categories. The results are shown in the next slides that we can see here. On the left, you really see the 4 categories of tumor shrinkage. You see patient with non-responder patients, patient with moderate response, fast responders, and fast responders with plateau. When we were looking at the exposures, the AUC steady state at 6 weeks, as we can see, there was no clear dose response. The effect was not significant. And what does this mean? That doesn't mean that there was no effect. That means really when we were looking at the results that we have on the left side of the slide, where we represent the proportions of progressive disease, stable disease, partial response, or complete response patients, we can see that the response or the proportions were pretty much similar across the different doses. And this type of results goes pretty much in line with what was observed with the biomarkers as well, suggesting that 10 milligram was obviously too high of a dose, and maybe getting at a lower dose of 2 milligram per kg might be helpful. And this is actually the dose that Merck decided to move forward. The next step, once we define a tumor growth model, is to relate the output of this tumor growth model with a clinical endpoint of interest. In our case, it would be overall survival. What we can see on the right is really on the left is the tumor growth profile and tumor size profiles that we observe together with the Kaplan-Meier plot. And what we want is to relate this tumor size with the probability of survival at every time. This is what is represented in the equations. That will be the only equations that we will have in this presentation. But it basically says either we relate to the tumor size at every time point, and we have a probability of event, or we relate to an endpoint, a derived PK matrix endpoint. As we have AUC or Cmax for PK, we can define similar derived PK matrix for the tumor size. And this is what we see here. On the right-hand side, there is standardized tumor size profiles, where we can see the 4 different endpoints. We have KS, which is a tumor shrinkage rate. There is KG, which is the tumor growth rate. These are the 2 rates that we defined previously in our model. And we have the early change in tumor size on those 24 weeks. We have the time to regrowth, which represents an idea. And all these metrics can be derived from the model and the individual data. These metrics can then be related to either OS or overall response rate or progression-free survival. This type of work has been done early on with the team of Clare et al. At Genentech together with atezolizumab to see what would be the impact of the metrics on overall survival. And they actually found a pretty good relationship. They found a significant relationship of the KG value or tumor growth rate on overall survival. What does this mean? This means that obviously the lower the KG, the longer is the overall survival. Similar conclusion. So this was done on one specific set of data, and FDA conducted similar type of analysis on non-small cell lung cancer patients with a larger database of 24 studies over about 10,000 patients. FDA came to pretty much the same conclusion, saying that KG was actually as well a good metric to predict survival, or at least to relate with survival, and that eventually we would need additional study to really look at the impact of this tumor growth rate to see how reliable it can be as predictors for efficacy. Now we were only talking about non-small cell lung cancer, and what Genentech and wanted to do, they actually extended the work on a larger database with different tumor types. They had the database of 10 clinical studies with 5 different tumor types: colorectal cancer, breast cancer, non-small cell lung cancer, renal cell carcinoma, and metastatic urothelial carcinoma. And they wanted, based on this database, they did exactly the same type of work and relayed the tumor growth inhibition model with overall survival. What they did is basically they estimated the tumor shrinkage and the tumor growth rate for each treatment and each tumor type, and they end up with different values for each individual. And then what they wanted is deriving the metrics of this TGI. So there was KG, there was KS, and there was other metrics as well as defined below, and looked at what was the most relevant one using either a semi-parametric approach, whether by a Cox model or parametric time to event. And interestingly, is that for these five different type of tumors, they actually come with the same type of results. There was a good relationship between the tumor growth rate and overall survival, and this is represented in the Kaplan-Meier curve that you see here, where you see the overall survival separated by the different quartile of KG. And as you can see, the lower the KG, the longer the overall survival. So without being saying that this is the Graal and this is going to be always true, I think it is really important to consider such type of endpoint to really look at the relationship between what we assess early on in our early clinical trial with what we can see in our late clinical trial, where we usually tend to only look at exposure itself. So to conclude this, I just would like to summarize and by showing a nice example that was presented, that was developed by Emily Schindler in the group of Uppsala University, where they actually did a complete framework to look at the effect of axitinib in metastatic renal cell carcinoma patients. They had a big database and they managed to relate the drug exposures of axitinib together with the biomarkers. Then they relate biomarkers to tumor size and ultimately to the overall survival. So they had a nice relationship for efficacy that we described all over this during all this seminar. And in parallel, they also managed to get the adverse effect relationship. So once you manage to develop such framework of modeling framework, it appears to be extremely helpful if you want to test different dosing, if you want to test different population. It can really be your decision making during the process. All this being said, maybe few take-home message for you. I think that it's important to keep in mind that modeling and simulation can play a role, and hopefully we demonstrated that it can play a role at different stage of the development, transition from preclinical to clinic, during the first-in-man, and after the first-in-man to support submission and exposure response analysis. We are fully aware about the challenges that remains in front of us. I mean, there is selection of patients, number of patients, heterogeneity, and early readout. I mean, this is something we are fully aware. However, we think that modeling and simulation, despite some of the caveats we know, can really help to make the decision, knowing the uncertainty there might be around and being aware of it, as long as we think about it early. So this is something we need to be planned. We really need to have this type of analysis within the development plan. We need to have this anticipated, and this needs to be done on an ongoing basis. We should really not wait to have all the data available. MNS is a tool that we should really use on an ongoing basis with update of the model to really help us to make some changes in the design or to adjust the design if needed, as we need to be more and more flexible. So with this, I really want to thank you for your time. Thank you for your attention. And I want to leave the floor for questions. We have few minutes for this. So Suzanne, I don't know if you want to take it back.
Yeah, thank you so much, Vincent. Great job to the team. Excellent presentation. And as Vincent said, I'd love to open it up to the folks on the line to pose their questions. So Julie, I think you're up with the first question. Someone wants to know, what's the best way to ensure the freedom to investigate informative endpoints in the clinical trial protocol and statistical analysis plan?
Yeah, so most of the informative endpoints are going to fall under the exploratory endpoints in any first-in-human protocol. And so those typically don't have formal statistical analyses associated with them in the protocol staff. and instead would be covered and reported potentially separately within the modeling and simulation analysis plan, which has a tendency to be more exploratory in nature and not require pre-specified analyses that are more than likely more rigid within a protocol-specific set. Now, remember, we're also in the first-in-human phase of development, so the staff of a first-in-human study versus like the statistical analysis plan for a phase 3 study, those are different levels of statistical analysis plan. So I think we also need to think about that as we plan for these analyses. You really, in the first-in-human setting and then this exploratory setting that we are in, should be leaving no stone unturned, regardless of what your statistical analysis plan has pre-specified for you to do in the first-in-human study.
Great, thank you. We've got a question on antibody-drug conjugates for our pharmacometricians. Do you have any clinical modeling and simulation examples for antibody-drug conjugate development programs?
So I think I'll take that one. So I'm aware there are published examples out there, and I would also suggest perhaps maybe looking through FDA reviews of some of the approved ADCs. One example I'm familiar with that has been published is belantamab mafodotin, and that's I believe developed by GSK. So that is another example where there was modeling and simulation used, and I believe it was based on just a couple of studies as well. So those are some examples. Thank you.
Thank you. I think our SimCip colleagues also, they worked with the folks over at Genentech. I think they used SimCip to develop a PBPK model for Polivy over there. Italo, can you talk to us about, were there any limitations to the phosphate-based simulations campaign?
Well, naturally, there are always limitations, especially in an accelerated program like that. Yeah, I think that a very clear limitation is that phosphate elevation was considered the only tolerability-related endpoint. So it was considered the safety endpoint to be controlled. And naturally, that is true if all the safety-related endpoints could be, let's say, were on target. Naturally, if we have off-target toxicity, that is not going to be valid, and this means that you need to have additional models. For instance, the one that Vincent showed in one of his last slides. There was, for instance, an independent relationship between plasma concentration and blood pressure, if I remember well, events. So that was a limitation. The interesting fact is that at the end of the day, FDA recognized the limitation, but it was they were accepting the conclusion that the dosing regimen was able to optimize the exposure to erdafitinib.
Great, thanks, Italo. Question for one of our metrics folks, maybe Vincent, you can take this. According to dose reduction and/or dose hold, dose reductions and dose holds often occur in oncology, especially under the maximum tolerated dose paradigm. Which PK metrics do you advise using regarding doing analyses on exposure efficacy or exposure safety?
It's actually a very, very good question, because that's a really big challenge, and we actually got this discussion internally about this, because that's really is a point. I mean, if we want to use a matrix, which is a stable matrix where we're talking AUC, we're talking average, we're talking C average, there is plenty of metrics we can use, and obviously they don't take into account all these elements. I don't have a clear, definite answer for this one. The best answer we have for this is eventually to use parametric modeling and really take into account all the concentration as much as you can. That's mean, for instance, if we're thinking about times to event, you want to use a parametric time to event, I think it would be more appropriate, because then you would take into account all the events. yeah, otherwise I think I would be tempted to use as much as I can and really try to test what is the best, but it would be more serendipity approach. Really no clear answer for this one. It's subject under discussion, I would say.
Absolutely.
Yeah, that's like a whole webinar in itself.
Yeah.
Yeah, maybe that'll be our next webinar.
Yeah.
All right, team, thank you so much. And thank you so much to our audience. It looks like those are the end of our questions. So on behalf of Certara, it's been great having everybody on this presentation. This concludes the webinar. Goodbye and have a great day.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye-bye, everybody.
Bye-bye.